My name is Alexa Feliciano, and I am a part of a team with Krista Leon for a biology project. After learning all the units of biology, we were tasked to connect topics from each unit to our final lab of the year regarding PTC tasting and the expression of the TAS2R38 gene. Starting with Unit 1, macromolecules are what each living organism is made of. The PTC lab focused more on nucleic acids, the hereditary molecules that define our genetics. Nucleic acids, also known as DNA, have adenine and thymine nitrogenous bases bonded together, and cytosine and guanine nitrogenous bases bonded together. Hydrogen bonds are what stabilizes the nitrogenous bases in DNA. To connect this unit to the PTC lab, hydrogen bonds within a double-stranded DNA molecule are separated through heating up the DNA to denature the structure. Polymerase chain reaction, also known as PCR, is what breaks up DNA in order to anneal and extend the single-stranded DNA strands to form a duplication of the initial DNA strand each repeated cycle of PCR. In Unit 2, we learned how each cell in our body has a permeable plasma membrane that is selective in what molecules enter, leave, or interact with the cell. In addition, the concept of surface area to volume ratio defines why cells are so small. Cells have a high surface area to volume ratio that allows easy diffusion of wanted molecules into the cell. Connecting these topics to the PTC lab, papillae contain the taste buds of gustatory cells, which are the receptors that molecules interact with on the tongue surface. Increased papillae on an individual's tongue means that there is an increase in surface area over volume, resulting in a strong reaction to detecting PTC. An individual with intermediate papillae on their tongue would still detect PTC, but someone with little to no papillae would not be able to taste PTC at all. Unit 5 was the unit of heredity, where we learned that DNA molecules make up the inheritance of genes. The main focus of the unit was meiosis, which is the sexual process of producing non-identical gamete cells to assort DNA alleles. Parents with certain alleles can pass down their genetics to their offspring through the independent assortment of genes, creating a multitude of various combinations of expressed genes in their offspring. These inherited alleles form the DNA genotype, which in turn expresses specific physical phenotypes for individuals. Connecting these ideas to the PTC lab, being able to taste PTC is having the hereditary trait of tasting receptors. After testing if we could taste PTC, each of us collected our DNA to analyze if we inherited the normal TAS2R38 gene in our genotypes. Unit 6 is when we learned how genes are expressed through the processes of transcription and translation of proteins. Transcription factors such as activators control enhancers to express certain genes during transcription, resulting in a phenotype being displayed. In the PTC lab, restriction enzymes were used to cut the target DNA sequence GGCC in the TAS2R38 gene. Non-tasters would have a mutant TAS2R38 gene where there is no GGCC sequence and is instead a GGGC sequence. That is why when analyzing the DNA of non-tasters in the gel electrophoresis had only one observed strand. Supertasters with the GGCC strand would have two strands due to the restriction enzyme cutting the DNA, and the normal tasters would have three strands due to inheriting a normal TAS2R38 allele and a mutant TAS2R38 allele. The bioethics topic I researched was on DNA sequencing and tissue engineering. Back in 6th grade, I came across the Vacanti mouse, where scientist Charles Vacanti grew a sheep cartilage ear mold inside of a mouse. Debates on this subject have been going on for years, especially since the discovery of no genetic modifications were done to the mouse during the experiment. The positive perspective on this topic is that tissue engineering can be a way to help reform damaged or deformed tissue cells in humans. By testing how cartilage can grow into desired structures, scientists can then use this method to help give tissue transplants to those in need, like ear transplants, for example. However, the negative perspective on this topic is the question if animals will be donors, meaning will we receive grown cartilage and tissue cells from the animals directly? In addition, DNA sequencing and gene editing is a part of tissue engineering, so there is a debate if your genome could be modified in order to produce reconstructed tissue cells in your own body instead of from animals.
Hi, my name is Krista Leon, and I did the PTC Tasting Lab with Alexa. So first off, we have Unit 3, Cellular Energies. With regards to the PTC lab, the main focus of this unit was enzymes and their functions, more particularly restriction enzymes. The binding of substrates to the enzyme's active site control reactions in the body. This is why they are biological catalysts. pH, temperature, and salinity all have the ability to denature enzymes. In this lab, we used restriction enzymes to cut up the DNA from our tongues so we could observe it. It's also worth mentioning that this unit covers cellular respiration, which consists of glycolysis, the link reaction, Krebs cycle, and the ETC, as well as photosynthesis, which consists of the light reactions and the Calvin cycle. Next, we have Unit 4, Cell Cycle and Cell Communication. The main focus of this unit with regard to the PTC lab was the signal transduction pathway of taste receptors. The substrate and the active site of an enzyme must agree with each other in shape and charge. Some people don't have receptors that can detect the PTC, so they don't taste anything. The components of signal transduction include the ligand binding to the receptor, transduction, and then a response. It is also worth mentioning that we went over different types of signaling, such as autocrine, paracrine, endocrine, negative versus positive feedback, as well as the stages of the cell cycle, which are G1, S, and G2, which these don't connect to the lab, but it's still worth mentioning that we went over them. Next, we have Unit 7, Natural Selection. So in this unit, the focus is on the 5 conditions of evolution, which are mutation, gene flow, genetic drift, non-random mating, and natural selection. The connection to the PTC lab is that the conditions of mutation and natural selection are at play with the TS2R38 gene. The ability to taste bitterness was actually a result of natural selection from our ancestors, the hunter-gatherers. This gene was useful to detect poison. Thus, natural selection favored those who had the gene because they could detect poison. It is also worth noting that in this unit, we also talked about Hardy-Weinberg equilibrium, different types of selection, and comparative anatomy. Finally, we have our last unit, Unit 8, Ecology. In this unit, the focus is on trophic levels and in the environmental conditions that sparked the expression of the TS2R38 gene. The fact that we even have a gene that can detect poison in plants suggests that humans used to be primary consumers. This mutation arose due to environmental pressures. Poisonous plants created fatal risk factors that our bodies adapted to avoid. However, not everyone has the TS2R38 gene. It is not common in those of European descent, likely because their environments did not have as many poisonous plants and therefore did not have environmental pressures to select for it. It is also worth mentioning that in this unit, we also talked about symbiosis, carrying capacity, and exponential and logistic growth. So the last thing I'm going to be talking about is bioethics. This company, Viagen Pets, can literally clone your dog. What they do is they'll harvest your dog's DNA by taking a skin biopsy, then they stir it in ice and save it until you'd like to clone your dog. Then they clone it by using somatic cell transfer and implant the dog's DNA into a surrogate. The new dog is genetically identical to the old dog. I can see why people might want this. It can help people cope with grief if their dog died. However, I personally disagree with this, and I think it isn't ethical. Viagen Pets has even stated that the new dog isn't exactly the same as the old dog. Sure, they look the same, but the new dog doesn't have the old dog's memories or experiences. Not to mention the procedure costs over $50,000. It's disrespectful to do this to a dog that died, and not to mention it deprives homes to dogs who are in shelters. It also opens this to a bigger picture. If dogs can now be cloned, how long until humans can? How would you feel if someone cloned you after you died? Well, thanks for watching.